opportunity to be able to introduce Iván Ortiz-Monasterio. He is someone who's been one of my heroes working in nitrogen all these years. I have been inspired by the work in Obregon. We're just looking on Google Scholar, 15,000 people have cited your work, Iván. I think that shows the value. I think the work around nitrogen efficiency, I know you've personally improved the planet, the environment that we all live in, because nitrogen is needed both to make sure we have enough yield, particularly high quality wheat, as you've worked even very recently publishing in that area, but also in terms of making sure that we don't exceed planetary boundaries. Nitrogen is unfortunately in excess in many places, and yet it's so scarce in others. And your work on nitrogen efficiency has led the way, people to better understand their plant, their soil, to target nitrogen. And we really appreciate you taking the time to reflect over your career. And I'm extremely excited to hear what you have to say today. So over to you, Iván, and thank you for all you've done.
No, thank you, Sig, for actually suggesting that I did this. As I mentioned earlier, it was actually fun to try putting everything together related to nitrogen. So thank you for this opportunity. So I'll start with a little bit of background. Here you can see a graph showing fertilizer use from 1965 to 2020. The blue bars represent nitrogen. And something that I found very interesting is that the biggest jump in fertilizer consumption occurred between 1965 and 1990. I joined CIMMYT in 1989, so basically the big jump in fertilizer use stopped when I arrived at CIMMYT. There was an increase of 4.1 times. Since then, since 1990, there has been an increase in fertilizer. Since 1990, there's also been an increase, but not nearly as large as the one that happened basically during the Green Revolution. Now, where all this nitrogen is being used, when we look at the consumption by crops, we see that wheat and maize use 38%, almost 40% of all nitrogen is used in wheat and maize globally. And when we look at the breakdown between developed and developing countries, we find that most of the nitrogen is being used in developing countries, 77% in this case. So this basically is where CIMMYT works. We work in the developing world in wheat and maize cropping systems. So we are positioned to have a significant impact in improving nitrogen use efficiency. There were two estimates on the efficiency of nitrogen used by cereals. The first one in 1999 by Bill Rahn, and he came up with the figure of 33%. A second estimate, basically 20 years later, by O'Mara, shows that there hasn't been much gains in the last 20 years in terms of efficiency in global use of nitrogen by cereals. However, O'Mara pointed out that there's actually a significant difference between countries in terms of efficiency. And we find here that the US, it's ahead of China and India. However, I'd like the work of Luis La Saleta, which he looked at basically 120 something countries, and he divided them in 4 different types of countries in terms of their nitrogen use. And he used here, yield refers to nitrogen yield. So he converted the yield of all the crops in these countries into nitrogen yield. And that's what you see in the y-axis. In the x-axis is the amount of nitrogen that was applied to the soil. And here you have the one-to-one line that would represent like perfect efficiency or 100% efficiency. So if you apply 100 kilograms of nitrogen and you recover 100, that would be like perfect efficiency. So the closer these color lines are to the one-to-one line, the higher the efficiency. And these colors represent different decades from 1961 to 2009. And the first type of countries are represented by India, China, and Egypt, where we see quite significant increases in nitrogen consumption and use, but a limited increase in yield. So again, India, China, and Egypt represent these type of countries that have had large increases in fertilizer use with an increase in yield, but not proportional to the amount of nitrogen use increases. The second group is represented by the US, Brazil, and Bangladesh. And here what you can see is that these countries started in a trajectory of low efficiency, but somewhere in the middle of this time period, they jumped to higher efficiency, to this trajectory of higher efficiency. Again, USA, Brazil, and Bangladesh represent these countries that through this period of time, they've been able to improve their efficiency significantly. Then the third group of countries, represented mostly by European countries, show that a reverse in the amount of nitrogen used. So they, for instance, here, France started with, I don't know, about 55, 60, but then sometime in the 90s started to dial back the amount of nitrogen applied by farmers. And the best example of all this, it's the Netherlands that went back to the levels of the 1960s. So a full return, a full 180 turn on the amount of nitrogen use. However, I think we need to point out that they started with pretty high levels to begin with, about 400 kilograms of nitrogen per hectare. So there was a lot of room to improve nitrogen use efficiency there. And the fourth type of country is represented mostly by African countries, where when you have these dots on the left side of the one-to-one line, represents crops that are extracting more nitrogen than the one that is applied in the field, therefore representing mining of the soil. The work that we've been doing on nitrogen use efficiency, it's in the Yaqui Valley. And the Yaqui Valley is representative of the first type of countries like India, China, Egypt. In fact, Oregon, it's agroecologically representative of environments where 40% of the wheat in developing countries is produced. And that's why it's so relevant to do research in this area, because what we learned here can be extrapolated to a much larger area in terms of wheat in developing countries. So when I first arrived at CIMMYT in '89, we put some experiments where here you can have different rates of nitrogen. The black circles represent putting everything at planting. And then the open triangles putting 100% at the beginning of stem elongation. This is about 45 days after planting wheat. And then only at the 225 rate, we looked at two splits. 1/3 at planting, 1/3 first irrigation, 1/3 second irrigation, and the other one 1/3 at planting and 2/3 first irrigation. And basically you can see clearly here that it's more efficient to apply the nitrogen later on compared to planting, and it's even better to split it. Not only in terms of yield, but also in terms of protein, higher protein by delaying nitrogen with respect to planting. And the simple reason for that is that here is the apparent fertilizer recovery when you apply everything at planting, when you delay it, and when you split it. So the reason why we get higher yields and higher protein by delaying or splitting the nitrogen is simply because we're recovering a larger proportion of the nitrogen that we're applying. So with that information from the on-station trials, we decided to compare the farmer's practice, which at the time was to apply 75% of the nitrogen pre-plant, nothing at planting, and 25% with the first irrigation. And what you can see here in the background is a nitrogen absorption curve for a spring wheat. And you can tell here that most of the nitrogen is applied even before planting the wheat. And the time here of highest demand of nitrogen is when they apply the least amount of nitrogen. So the new practice recommended nothing pre-plant, a little bit of planting because the requirements at the beginning are low, and most of the nitrogen at the beginning of stem elongation when you have the big demand of nitrogen. And we put 30 trials in farmers' fields to test these two systems. And overall, we found that the 1/3, 2/3 avoiding pre-plant applications gave higher yields, but it was even more consistent and the impact was larger on grain protein by trying to match better supply of the fertilizer with the demand by the crop. And again, when we look at the apparent fertilizer recovery, we get higher yield and higher protein because we're recovering a larger proportion of the nitrogen that we apply. So with those 30 trials, we measured that the fertilizer recovery following farmers' management was only 31%. So what was happening to all that nitrogen that was not recovered by the crop? And so here you see all these different pathways of possible losses. And that's when we started a collaboration with Stanford University. Actually, we started the collaboration with Berkeley because Han Matson was working at Berkeley when we started our collaboration. And the idea was to try to understand where was all this nitrogen that was not recovered by the crop going. So we compare farmers' practice, which was this 75-0-25 with 200 kg of N, versus the new practice of 1/3, 2/3 at a lower nitrogen rate. And we measured nitrous oxide emissions, and we found that we could reduce emissions by 50% without affecting yield or quality. And this got published in Science in 1998. Then one of the students from Stanford, by then, Han Matson had moved to Stanford University. One of her grad students was tracing the amount of nitrogen in the drains that were coming out of agricultural fields like here. And he found a range of values between 0 and 12 for nitrate and 0 and 12 also for ammonium, but with an average value of 6 milligrams per liter. So another student, Mike Beeman, he used 5 years of satellite images looking at the Sea of Cortez. And he demonstrated that every time that farmers fertilize and irrigate wheat here in the Yaqui Valley, it coincides with an algae bloom in the Sea of Cortez. Of course, we know that these algae blooms have very negative effects on the marine life. And then we did also a study of the amount of nitrates in well water. This is the Yaqui Valley, and the squares represent the location where the water was sampled in these different wells. Most of them were within the allowed levels of nitrate, but there were already a significant number of sites that were above the recommended levels, and there was one site 10 times higher. So I always use these three images when I give talks to farmers here to tell them that the decisions they make of how much nitrogen they're going to apply in their field has consequences well below their fields at a global scale because of the nitrous oxide emissions, at a regional scale because of what's happening in the Rio Cortez, and at a local scale because of nitrate contamination in their aquifer. So here is a diagram showing when farmers apply 250 kg of N, how much is taken up by the crop. We looked at leaching and two studies with a little bit of different results. One measured between 2 and 5% losses due to leaching, but another one 14 to 26. Then 2 to 5% lost as NOx and nitrous oxide, 20% immobilizing the organic matter. What we didn't get to measure that somebody can follow up on this is what happens to ammonia volatilization. We believe that's one of the main pathways of loss. And also the non-exchangeable ammonium, because we have vertisols in Obregon in a significant part of the valley. These are two-layer lattices that when they're wet, they expand and they can trap ammonium between the lattices. So we think that's also a significant amount of nitrogen that can be immobilized temporarily in those positions. So we collaborated between 1992 until 2012, which is when this book was published, that summarizes all the work that we did during this period of time. So in this study, the previous study showed that rate was one of the key factors to be able to improve efficiency. And identifying the right rate presents two challenges. The first one is to figure out how much nitrogen we have in the soil. And this graph represents 28 farmers' fields where we measure available nitrogen, not with a soil test, but by allowing the crop to absorb nitrogen in areas that were not fertilized. And you can see that it goes from below 50 to more than 200. So this is a challenge to be able to accurately predict how much nitrogen you have in the soil. So that's on the supply side. On the demand side, here in the valley, we have shown that yields can vary from one year to the next between 5 and 7 tons. And of course, the demand is going to be much different in a high yielding year than in a low yielding year. So we actually did a study with a model of farmer management where we introduced climate data and soil data. And we found in this study that being able to predict accurately the amount available in the soil was 2 to 3 times more important than quantifying if it was going to be a high yielding year or a low yielding year. So again, being able to predict the right rate, it was a big challenge. So that's when we started our collaboration with Dr. Bill Ron at Oklahoma State University. They were developing these rigs that had sensors and variable rate application. So our collaboration was, how can we adapt this technology to smallholder farmers in the developing world? And from that time on, we started getting a stream of grad students. Here you can see the prototype of the Green Seeker being tested by an OSU student here in Obregon, which became the first version of the Green Seeker. Then we established this type of trials to calibrate the sensor for use as a diagnostic tool. Here you have different nitrogen rates, different timings to generate different NDVI values, which is what the sensor is measuring, and correlating those to yield. And you get this type of graphs that represent the calibration of yield versus InSafe, which is NDVI divided by the number of days between planting and sensing. And we developed 2 algorithms for Obregon. One is in the Oklahoma State Nitrogen Use Efficiency webpage. You just need to introduce the NDVI of the enriched strip, which is, actually I should go one step back. The way the technology works as a diagnostic tool is you need to establish a reference strip. A reference strip is a strip in the field that has a non-limiting amount of nitrogen. And you use that to compare with the rest of the field. And you do the testing in the case of maize around V6, and in the case of wheat at the beginning of stem elongation. Then you put that data in these algorithms, and it comes up with the fertilizer recommendation. But I would like to stress that this enriched strip is a technology on its own. It can be very helpful independent of the sensor, because here you have a strip that you know is not limiting. You compare it with the rest of the field. If the difference is small, then you know that you only need a small amount of nitrogen. If the difference between these two areas is large, then you know you need a large amount or a medium amount. So it's a technology on its own. The sensor helps to make it more quantitative. So once we had the calibration, we were ready to start testing this in farmers' fields. Cesar Valenzuela, which was our neighbor, just on the other side of the fence at the research station, he told me, okay, I'm ready to try this sensor. He had 100 hectares. He applied 11 tons of chicken manure. His enriched strip was 22 tons of chicken manure. We went with the sensor that recommended 0, which meant he didn't have to apply any more nitrogen. What he ended up doing was he applied 75 kg of N in half of the field and nothing in the other half, and he got the same yield. So we were then ready to move on with other farmers. It started kind of slow. You know, this is the adoption area of the use of the green seeker in two valleys, the Yaqui and the Mayo Valley. And we started like with 3 farmers, 5 farmers, 8 farmers, until we started collaborating with AOAS. And AOAS is the umbrella organization for 7 farmer unions. And they said, okay, you know, we will support the transfer of this technology to these unions. And we got the support from the government to pay for the farm advisors that transfer this technology to farmers' fields. Daniel Lapidus, who's more of an ag economist, we shared with him all the data that we collected from these farmers' fields, and he summarized it and published it here. Basically, he found that $37 was the savings for the use of the green seeker, and this was the amount of CO2 emissions that were avoided, and the technology was used in 48,000 if we sum all the 8 years that was used. Then we decided to move this technology to other parts of Mexico, like Baja California and Guanajuato. We also shared that data with Daniel Lapidus, and there, in the case of wheat, the savings were $44, and in the case of Guanajuato, $51 for adopting the technology. We continue collaborating with ASU. In this case, first they were calling it the pocket sensor. This was the prototype that we tested with farm advisors in Yaqui, but also students from OSU will be coming every cycle and collecting data. But when the farm advisors used the prototype, they said that after pushing a button here for more than a minute, their fingers were cramping. So they suggested that the sensor should have a handle and they should have a trigger. And the company liked the ideas very much, to the point that they implemented those suggestions from the farm advisors into their final product. So it's just Jerry Crane, OSU. He's the person that demonstrated that the big NDVI sensor measured the same thing as the smaller NDVI sensor. So this collaboration with Oklahoma State University was very productive, and we received many groups of students every year that would come here for about a week to learn about agriculture in areas like Obregon, where the Green Revolution started. Then at the beginning, so our goal was to find cheaper versions of these green seekers that could be used by smallholder farmers. And then we started testing other platforms where you could also measure NDVI, which is the same thing that the green seeker measures. And first, we started looking at these manned airplanes more for research purposes. And here we were able to mount a hyperspectral scanner. And we had a trial where we had 2 types of tillage, 2 types of irrigation, and 6 different ways of managing nitrogen. And we collected these hyperspectral data, trying to get, better assessments of yield, total N uptake by the plant, and grain protein. Grain yield, we were able to get R squares of about 0.6, and for total nitrogen uptake, R squares of about 0.7. So I would say, fairly good estimates of these 2 traits by using remote sensing. But what has been very elusive is grain nitrogen. Here you see different vegetative indices. And, I think that once we looked at all 4 years, I think the best R square we could get was about 0.2. So, this is a challenge that remains to be solved. How can we more accurately measure grain in the nitrogen in the grain, which would be of great value for farmers and for the companies that are concerned about having high protein wheats. So then here with my colleague Urs Schutles, funding from Masagro, we work on developing this GreenSat. We collaborated with CIEP, which was part of Sagarpa at the time, which is SADER now. They manage an antenna for a French satellite called SPOT. So they bring down all the data from the SPOT satellite related to Mexico. They agreed to share with us NDVI maps for several regions of Mexico. Here you can see the NDVI map for the Yaqui and the Mayo valleys. And the tool actually worked pretty well. Here is again an NDVI map of the Yaqui Valley. These squares are the typical 2x2 squares in the Yaqui Valley. I'm gonna make a close-up on this white area. But previous to all this, we looked at the NDVI from the Green Seeker versus the SPOT satellite, and we got an R square of 0.7, which I thought it was satisfactory. And again, this is the close-up of that white square. This would be the enriched strip. So the way the tool worked was you would click here, that would allow you to draw the perimeter of the area, and automatically would add up all the pixels for NDVI and put them in this box here. That would be, this was the nitrogen calculator. Then you go to the other area where you want to make the diagnostics, draw the perimeter, and again, it puts the NDVI average here and automatically calculates how much nitrogen you need to apply. Did the same thing on the other side, calculates the NDVI for that area and gives you the nitrogen recommendation. So it was all very good, except that the first priority for this data was national security. The second one was natural disasters. The third one was they had to generate a full map of Mexico every so many months. And then the fourth priority were other projects, and we were among other projects. And the problem was that we were not receiving those images at the critical times. So then we started working with drones. we started working with this EV, and we really like this camera called Sequoia that also allowed us to measure NDVI. Here we see our team flying the drone while another person is measuring NDVI with the green seeker. And we found, in the case of wheat, very good relationship between NDVI from green seeker versus NDVI from the Sequoia camera. And the way we did these calculations was, here we have 3 images, one with the EV drone with a resolution of 11 centimeters per pixel. Then with the airplane, it was 50 centimeter pixels. And with the spot satellite, it was 5 meter pixels. So what you see here is the trajectory that followed the green seeker. So we were sampling this square with the green seeker, and then we average all the pixels from this area of the spot satellite, and that's the data we use to make this comparison. And here you can see that with the EV, you can distinguish the individual trees compared to the satellite that doesn't allow you to give you that level of detail. Yeah. So this last crop cycle, the hub presented a project with FIDA, which is a funding agency for technology transfer, so that they could hire farm advisors that could transfer and use efficient technologies to farmers' fields. And this year, the hub was able to transfer this technology to 6,000 hectares. And I will talk in a minute what that technology included. And also the Farmer Association, AOAS, adopted some of this technology. So the hub transferred 3,000 hectares and AOAS transfers 6,000 hectares of technology that included improvements in the rate, the timing, the source, and the placement. And I'll detail here. This is the current farmers' practice. They still apply 55% of their nitrogen pre-plant, nothing at planting, 25% first irrigation, and 20% second irrigation. So the new practice is no pre-plant application, only a little bit of the nitrogen at planting and most of the nitrogen here, with a little bit of nitrogen in the second irrigation to avoid the problem of yellow berry. And here, the recommendation is not to apply the nitrogen in the water, which is what they do, but to apply it solid. So these 2 changes represent improvements in rate, timing, source, and placement. Here, you can see the use of the drone. Again, resolution of 11 centimeter per pixel. Here's the enriched strip. And AOAS this year started experimenting, not using the enriched strip, but using a proportion of the field that had the highest pixels with NDVI as a substitute of the enriched strip. And they found a good correlation between a proportion of these high NDVI pixels in the field in comparison to the enriched strip. Now, the other big change that has occurred since I first started working here is I thought that it would be very easy for the farmers to stop applying nitrogen pre-plant. But they couldn't do it because they didn't have the machinery to be able to apply it at planting or later on, until very recently, where this type of spreaders have become available in the market here. See, the challenge was that when they apply their nitrogen pre-plant, they could use this very large equipment that could, they would fertilize 15-meter strips very rapidly. But this equipment makes 30-meter strips of fertilizer. It's even faster than the previous machinery that they were using. So thanks to this improvement in the availability of machinery, now they are finally starting to switch from avoiding pre-plant applications to making their first application at planting. Another fruitful collaboration was with Phil Robertson and Kevin Kackmark, Kellogg Biological Station, which is part of Michigan State University. This is Phil and Kevin. Neville also closely collaborated, but he's not in the picture. You know, they've found, you know, back in 2007, 2008, in 6 maize trials, they were finding that as you increase the amount of nitrogen, that nitrous oxide emissions were not linear. At that time, IPCC was saying that this increment in fertilizer rates should be considered as a linear response. So first they started seeing that it wasn't really quite linear. So based on this information, they decided to do a meta-analysis of 82 trials around the world. And they found, you know, this red line is the IPCC. The dotted line represents the maize trials, and the solid black line represents the meta-analysis. So they pretty much demonstrated that it was exponential. It was not a straight line. Now, the very interesting thing was that among those 82 sites, there were no trials where either maize or wheat had been tested in the tropics or the subtropics, which is where CIMMYT works. So we found that knowledge, that gap in knowledge, and that's when we established these 2 years of trials in Oregon, where we again confirmed that the response was exponential and not linear. And now we have the exact emission factors for wheat here in northwestern Mexico. So I've also worked with the breeders on nitrogen use efficiency, and I'd like to present some of those results. When I first arrived as a postdoc, Ken Sayer has started a trial looking at these different genotypes with these different nitrogen rates. So I arrived the 3rd year to complete the 3rd year of that. And my main contribution was to bring the perspective of Moll et al., which was suggesting that the nitrogen use efficiency for genotype should be evaluated looking at uptake efficiency and utilization efficiency. So here we have this represent the year of release of the varieties. So we could measure genetic gains over time at these 4 different rates of nitrogen. Now at the 300, you can kind of see that it's a steeper slope, but this is an artifact of the fact that this variety, Yaqui 50, was a tall variety, and with 300 kilograms of nitrogen, it lodged. That's why the yield came down. So what we did was then eliminate Yaqui 50 in this analysis, and we found that basically all 4 lines were parallel, so gains were the same. And at the time, the big discussion was people were saying that Green Revolution varieties did not perform well on their low levels of nitrogen. And here we could clearly show that yes, there were genetic gains, and in relative terms, they were as high as the ones with different rates of nitrogen. Then we did a trial that took 10 years with my colleague Martin van Ginkel, because somebody else said, oh, but you already selected those varieties on their high levels of nitrogen. If you had selected them on the low levels of nitrogen, you would probably get a different result. So we selected 2 good uptakers and 2 good utilizers of nitrogen. He made the cross, and he selected from F2 to F6, always on their high nitrogen. Another group of that cross was divided to be selected always under medium, another one always under low. A fourth group was high in Yaqui, low Toluca, high Yaqui, low Toluca. And the last group was low Yaqui, low Toluca. And these were evaluated. The best lines that came out of these selections histories were evaluated at 3 different levels of nitrogen. And the results show that the best regime for selection was high Yaqui, low Toluca. That gave the best yielding lines for 150 and 300. And the worst one was alternating low Yaqui, high Toluca. And I think the reason was that I remember one time asking Dr. Bola why he didn't select under low nitrogen at the beginning of the Green Revolution, because many farmers were not using fertilizer yet. And he said, under low nitrogen, I cannot distinguish the good genotypes from the bad genotypes. And I think that's why we see that this was the worst environment, because Yaqui is where breeders select for yield, Toluca is where they select mostly for disease resistance. And then under low nitrogen, all selection regimes showed progress, and there were no differences among regimes. So if you were going to grow them under low nitrogen, it didn't matter which of these schemes you would use. You found progress in all of them, but there were no difference between selections regimes. I'm almost here to finish. And the biological nitrogen inhibition, I was fortunate to participate in this project, where the idea is to bring genes from a wild relative of wheat that generates root exudates that inhibit nitrification. And some preliminary data shows that this could reduce nitrous oxide emissions. This needs to be confirmed in larger field trials. But I think it's an innovative technology that looks very promising. And just to finish very quickly, I think there's also an important message on wheat quality and nitrogen use efficiency. Here we find the grain yield of this variety Cirno, and we find that the yield increases up to about 160 kg of N, and then it becomes flat. And this is yellow berry, which would be an estimate of, let's say, protein in the grain, but we want low yellow berry. And we find that to be able to get the lower yellow berry, we need to add additional nitrogen that is not going to improve yield, but it's going to improve the quality. And the French have found the same thing. Here is grain yield, nitrogen applied, and here they found, these were 15 trials they did. They found that the optimum rate for yield was about 220, but this is grain protein and nitrogen rate. They found that to be able to obtain the protein that they want, which is 11.5, they need to apply more nitrogen than the one that they applied to optimize yield. And is this amount of nitrogen the one that causes the bigger problems with nitrate leaching in their case? And what we have found is that the emissions of greenhouse gases become exponential once you start applying more nitrogen above the optimum for yield. So I'll put an example here. So the main variety used to be Hupari here in the Yaqui Valley. This is the yield, and this is this yellow berry, which is affecting quality. This variety had no problem with yellow berry, but had lower yield. When this one became available, then it was widely adopted, but it was very susceptible to yellow berry, which meant that in order to bring it here to a level of acceptable berry, yellow berry, farmers had to apply additional nitrogen above what they needed for yield. So many conclusions on agronomy. I think there's still a lot of room around the developing world to make significant improvements in nitrogen use efficiency by using the 4 Rs. I think precision agriculture will be slow to move into developing countries, and it will have a bigger effect on efficiency than on yield. Being able to predict nitrogen with remote sensing remains a big challenge. I've also found that trying to transfer this technology to farmers' fields, we could benefit if there would be policies like they were implemented in Europe that would push farmers a bit more to adopt nitrogen use efficiency practices. And something that I've always believed is that we need to find ways to bring back the livestock systems into the crop systems so that all those nutrients in the manure can be more easily recycled. And on the breeding side, I think that it will be interesting for somebody to look at the environmental cost of producing high quality wheat. And I think this information, if verified and confirmed, could be used to develop a project for improving the type of protein rather than the amount of protein as a way to improve grain quality, which is environmentally friendly. And just finally, saying that BNI, I think, is a promising and innovative approach that should be pursued by CIMMYT. Thank you.
Thank you so much, Iván. I think there's so much. Can you go back to your conclusions there? Maybe people want to see that slide. Yeah, I think that's really great. So please, everyone, this is open to questions. What would you like to learn more about this really amazing tour of nitrogen, tour of force? Thank you again. Maybe we do a quick hands. It's always challenging to do online, but really appreciate that. Any questions?
Yes, Juan.
Yeah.
Oh, sorry a second.
Hi, Iván. Very presentation. Good to see you again. I have a question. In the result for this work, you use different nitrogen levels to do selection, yeah?
Right.
In which when you started with the Shaki in low nitrogen, you mentioned it was the worst. My question is, how much makes sense, or what do you think really about to do selection in on-farm trials that usually they have low inputs, low nitrogen, no irrigation, probably other not so good agronomical practices?
Well, I would have two comments. Like Borlaug said that he didn't want to select under low nitrogen because he couldn't distinguish the good ones from the bad ones. So I think you need to then look at the approach of selecting in farmers' fields, if that is going to allow you to select the good ones from the bad ones. If that's going to mask that ability to distinguish them, maybe it's not such a good idea. That's on one hand. On the other hand, selecting on their farmers' fields, like for instance, if weeds are a problem, then you could probably benefit from selecting types that are more competitive to weeds. So I guess I'm not giving you a straight answer. I'm telling you that I see advantages and disadvantages of doing it.
Okay, thank you. I know it's not an and there is no an easy answer. No,
Sure,
it's
right.
more complex. But thank you.
Sure.
Like lots of hands, Janet, and well, Debash, you were first, Deb, and then Janet.
Hi, Ivan. Yes, it's a really very impressive. I must say, it's a very eye-opening, lots of learning. So thank you very much. I have two very specific questions. One is that how we can reach to the agronomically optimum nitrogen. Is it a very site-specific, or we can have say, at least as a region basis, a broad idea about what should be the agronomical optimum? What should be the approach? And the second one is that what is this to increase the nitrogen use efficiency, the agronomic approach and you have the breeding approach. So are they very complementary, or one is much ahead of others? Thank you.
Sure. No, thank you. Thank you for your question. I think we know very well what's a good management practices in terms of improving nitrogen use efficiency. And I think the 4 R's, right rate, right time, right source, and right placement is the way to go. You need to optimize those 4 R's for your cropping system. And as I said, I think we know a lot of what's the right thing to do in many cases. In other ones, we will need to establish some trials to answer that, but I think we probably know most of the answers. So I would say, try to apply the 4 R's to optimize nitrogen use efficiency. And I think there's more room. So what's more important, breeding or agronomy, to improve nitrogen use efficiency? And that's another difficult one. And when you look at papers that look at the progress in yield over time, there's an agronomic component and there's a breeding component. And nobody really has measured it, but they say, well, it's about 50-50. Maybe to keep everybody happy, I'm not sure why, but it's something difficult to measure. But I think both, probably I would say both are as important. Because as we increase the yield potential of the new varieties, we will be improving the nitrogen use efficiency of the system. And then again, something that I'm realizing later in my career is that quality is probably playing a very important role that nobody is really thinking about, I think. And it's again that some of these varieties would need significant amounts of additional nitrogen to obtain that protein, that quality that you need, and others don't. So we should be looking very closely at that, trying to select those varieties that don't need a lot of additional nitrogen to obtain the protein that we want. So again, I think I would probably finish by saying that both are important, probably as important.
Okay, thank you, Ivan.
Iván, thank you so much for your talk, and congratulations on all the progress that you've made. It's really amazing. Yeah, I think you kind of addressed the question that I had regarding you had made the suggestion about protein that might be more environmentally or climate friendly in terms of breeding. And it sounds like I think your slide mentioned a type of protein, and I wasn't sure what you meant by the type of protein, but you've just kind of elaborated a little bit on
Sure.
Protein quality at the end in terms of nitrogen responsiveness.
Right. Yeah.
It sounds like, yeah.
So there's 2 ways to improve quality, right? One is the amount of protein, and the other one is the type of protein, gliadins versus glutenins, etc. So I was referring more at looking at gliadins and glutenins to improve quality rather than improving the total amount of protein. And,
okay. Yeah. The end use quality versus, yeah, gotcha. Mm-hmm.
Something that I skipped this one because of time, but this is the amount of nitrogen that different varieties need to obtain the 11.5 that the French need. And there's some varieties that need, between 0 and 40 kilograms of nitrogen, while others need substantial more amounts of nitrogen to obtain that level of protein. So we should be focusing on selecting this type and avoiding releasing this type that will require increasing the total amount of nitrogen that is applied in the cropping system.
This is really, this penultimate slide here, I think is very fascinating. And thanks again for opening up our eyes to nitrogen use efficiency, to your deep knowledge on these areas, to, right, as it said in this chat here, thank you, Kendi, amazing career. Thank you for sharing a rich experience, Iván. This has really been eye-opening for many of us, even someone like me who's worked in nitrogen for many years. I think it really shows there's always something new to learn, and that, you know, that evidence-based approaches to science really bring so much. And I know it is time and that many people need to move on, but I'm very pleased to see that Isabel has shared the live stream, and we will definitely be making sure that's available to everyone, including many of our partners. So thank you again, Iván, for this very instructive and interesting presentation. Really appreciate it. Taking the time. Pause and reflect.
No.
We need more of that. Thank you so
Thank
much.
you for the opportunity, Sig. As I said, it was fun to put all this together.
Excellent. And I know people will be continuing to contact you, I'm sure, for, you know, to keep learning from you on this. And yeah, thanks again for that. Really, we're going to miss you. It's and you know, hopefully gauge you in various ways, because this is shows again what it means when you can look back over a career that has really focused and really provided unique insight. Thanks again, Iván,
Yeah.
and we'll continue try to keep this work going. Go ahead.
Something that I should have mentioned, that I meant to mention, I forgot, is really the importance of good collaborations. I mean, I was very, very fortunate to be able to collaborate with great people. And it was thanks to them, to those collaborations, that I was able to, you know, to do what I did. So finding good collaborators, very, very important.
Very good words of advice to the young scientist here in this talk. I agree. I could agree more. Absolutely. And of course, I think a lot of Phil Robertson, having had the privilege to work with him as well. So nice overlap there. Great. Thanks again. And nice to see such a wide range from remote sensing to ag economists to our regional representative